Now, thinking about the first problem that we just mentioned there in terms of metabolic instability, which can lead to high clearance or low oral bioavailability, typically we would assess this in microsomes. And so typically we might look at the intrinsic clearance, half-life, or indeed turnover in human liver microsomes, which would high in turnover clearance or short half-life typically would indicate rapid metabolism by cytochrome P450s. And if we observe this, we can use in silico models to help to guide that optimization of compounds to improve their metabolic stability. So, for example, in silico models can identify the sites of metabolism, otherwise frequently known as metabolic hotspots, and give us guidance on where we need to block that metabolism to improve stability. And if we look here on the right-hand side, we can see the predicted regioselectivity of metabolism by the major isoforms responsible for metabolism of this compound. And you can see that it's likely to be hydroxylation on these different methyl groups will be the predominant sites of metabolism. But that in itself is important information, but not sufficient to really guide the optimization of these compounds. An additional piece of information we can provide is what's known as the site lability. This is an absolute measure of reactivity to P450 metabolism. So we look at this in terms of this metabolic landscape, where the different sites are sort of ranked in terms of their lability or the vulnerability of those sites to metabolism. And we can sort of summarize this in a value called the composite site lability, which is a measure of the efficiency of product formation and hence an increased risk of rapid clearance. And so actually there's more information. There is actually a third highly labile site here on this compound, which although it's responsible for a small proportion of metabolism, in absolute terms, it is also highly labile. So if, for example, we were to modify or block these predominant sites, what we're likely to see here is metabolic switching to an also highly labile site and hence see a relatively small decrease in the overall rate of metabolism, i.e. we have more work to do on this compound. Now, it's important to note that this site lability or composite site lability is not a prediction of the overall rate. It is one very important factor, but other factors are important. For example, and not least binding affinity. If a compound doesn't bind to a P450, it doesn't matter how labile the sites are. And we'll come back to that question in just a moment. But first, I thought it would be interesting to look at a specific example of this to show how one can use this in practice. And I'm just going to show a very brief and simplified version of a study that was published actually using much older versions of the models that we used back in 2004, and it was published in Bioorganic Medicinal Chemistry Letters. But this focused around the design of analogues of Buspirone. So Buspirone is a drug. It's an anti-anxiolytic and a ligand for 5-HT1A. It's got excellent receptor affinity, 25 nanomolar, but it does suffer from poor and variable oral bioavailability, and that is driven by metabolism by cytochrome P450, specifically CYP3A4. And one can see this in vitro. It has a half-life in vitro of 4.6 minutes. And so the goal of the project was to identify analogues of Buspirone with significantly improved CYP3A4 stability, in this case aiming for greater than threefold improvement, so roughly 15 minutes, but also, of course, retaining receptor affinity, and the project was willing to sacrifice about an order of magnitude. And in terms of the experimental data that's available, here we're highlighting the primary sites of metabolism that were identified experimentally, so the para position on the pyrimidine, the alpha position to the nitrogen in the piperazine, and also this spirocyclopentane here have been observed experimentally. And so let's actually do this in practice just to show you what this looks like. So I'm just going to jump, in this case, into our StarDrop software. And here you can see I just have Buspirone and a couple of analogues. We can easily calculate the predicted metabolism by cytochrome P450s. So let me just do that. This actually appears instantly, and just to be completely transparent, these are pre-calculated results. They're stored on the server when you calculate them to save time and not reproducing the same results multiple times. In practice, that might take a few minutes to perform that calculation. But this here is Buspirone itself. And just really to show you here, this is the predicted regioselectivity of metabolism, in this case by 3A4. We'll talk later about which enzyme in which P450 model, so I'm just going to hide those away for the moment to focus on the metabolic landscape and the sites of metabolism. But what you can see here, again, rather nice to see, is those predominant sites of metabolism on the pyrimidine and adjacent to the nitrogen on the piperazine are indeed predicted to be the major sites of metabolism. And although it's a minor site, you can also see here on the spirocyclopentane. So, this is giving us a good representation of what's happening experimentally. And so what we can see here, there are two highly labile sites, again, on the pyrimidine and at that alpha position there. Those are the sites we would want to focus on, but maybe also looking at this end of the molecule here. And what you can see are two other analogues that were designed to address some of these questions. We can, again, select those and calculate their metabolism by cytochrome P450s. And again, seeing the results. We can see here significant improvements in that metabolic landscape. There are no highly labile sites. Here you can see we've blocked that para position here and added some steric hindrance there. And if we look at the third example, the second example here, you can see actually making modifications again with a fluoro, blocking that site, and at the changing that spirocyclopentane into a gem dimethyl, also significantly reducing that metabolic landscape. And that is also reflected in this composite site lability that you can see summarized here. So driving that composite site lability down has a significant effect. And when these were tested experimentally, you can see indeed, in this case, a half-life of almost 80 minutes. And here again, achieving that goal of about 15 minutes while retaining good activity against the 5-HT receptor. So you can see how we can find these sites, provide that guidance to blocking those sites to improve their metabolic stability. And so just to summarize those results here, you can see removing those highly labile sites, achieving a significantly lower composite site lability, indeed increases that metabolic stability. Here again, you can see a couple of other examples where we can significantly increase that half-life in vitro.